So this is going to be a little bit different than what you got from Ignacio. This is actually going to be drilling down into actually what's going on in the soils. And this study was a metagenomic analysis of USA, Europe, and Brazilian soils for a crop protection product safety study. So if most of you in ag, you realize that degradation studies for chemical compounds for soil application are routine in the ag industry. We spray things out there and we need to know what happens. That's what the EPA for and FDA and places like that are. We completed a metagenomic population analysis of the soils from 3 different geographic regions for soils that are known to be fast degraders of these herbicides. The goal here is to see if there's any similarities between the soils to account for the fast degrading phenomenon. So a little background like Ignacio gave us. So, metagenomics and there's metatranscriptomics. And prokaryotes represent millions to billions of separate genus species in the environment. And the second point, the 0.1% to 1% of these organisms can be cultured by conventional methods. That's been bandied around for years. We were doing this in the 90s, and they were talking about the same number then. And people say you can actually get up to 10% culturing, but as he was just saying, we don't know we have no idea actually how much is even in there. Is there, there's probably way more than we're estimating, so that number might be even, a lot less than those numbers we're talking about. And just as a gram of soil can harbor 1 billion microbes of 4,000 to 7,000 species, it's big numbers. And metagenomics, which is the DNA, and metatranscriptomics, which is the RNA, bypass the need for isolation and cultivation of these microbes. So an overview of what we did, we collected samples from 19 soil samples from untreated regions of the fields. And these soils from the USA, England, Switzerland, and Brazil. And we had 3 5-gram replicates per sample, and a total of 57 samples were analyzed. The DNA was extracted and converted to index Illumina-MiceSeq libraries and run in groups of 6 to 8 per run. A BLASTX similarity search was performed, and the data was analyzed by the program Megan. Hopefully I'm not getting too deep in the weeds for some of you. I don't know what everybody's background is, but I try to keep it in the middle so we can, for those who want more, we'll have it. And for those who didn't want to hear all this, hopefully you'll survive. It's only 10 minutes. So some Megan basics. So Megan uses the lowest common ancestor algorithm to assign the reads to taxa. So what that means is a read with hits passing a threshold of 3 different species of streptomycetes, but no other genus, will be assigned as the streptomycete genus, not one of the species. So the program will push things down as far as it can toward the best series of matches, and if not, it kicks it up, keep kicking it up all the way up to back up to the phylum if that's the best place that it fits. And Megan assessments are relative. Assignment to a particular taxa should be interpreted as I am most similar to, not I am. Because remember now, this is BLASTX. We're looking at protein, DNA converted to proteins. And this is particularly true in taxa that are more distant from the root of the taxonomic tree. And Megan can be used for taxonomic, pathway, and functional characterizations of metagenomic samples. It's a very powerful program, if any of you are familiar with this. And why we like Megan. First, we like the protein similarity match, which we believe is more representative of what's there versus 16S. And the second reason is it allows us to go back to the same library and sequence deep if we see something that's interesting, without creating a new NGS library. Because there are cases where you get a sample, and that DNA sample might be the only one you have, and you don't want to be going to it very often to be looking for other stuff. So if you can make one library and use that to capture everything, that is very important in your analysis versus going in to 16S and doing something and then have to go back if you want to go back in and look what's going on at the protein or a gene level. So we have it here. This is the United States soils, and we used 250,000 reads, paired in reads, and these were placed at the class level. So all this analysis is at the class level. If you remember your kingdom, phylum, class, order. Well, there's a mnemonic for that, but I can't remember what it is right now. But thinking about this is what we found was very interesting was, which was very helpful for us, is the replicates clustered. We were very concerned about going into a sample and taking something out and have one little dirt clod in there. It could be a super microbiome by itself, and it could be just all, let's say, Bacillus thuringiensis, and it's going to skew everything beyond what you want. And the other thing is, when you look at these, is you can, oops, that's the wrong one, go back, is that this is from 2012, and this is the same field from 2011, same here, and same here. So you see seasonal variations among the population in this area. And what's interesting is they stay clustered together even though you're losing something and gaining something over that period of time. I thought that was pretty interesting. That was very, very helpful for us to be able to help with the analysis and taking things forward. This is all the Brazil soil samples, and I present this as percent counts, remembering that we're using 250,000 reads for each of these sample analysis. But keep in mind, as Ignacio said, we're finding about 20 to 30% of the reads don't match anything in the current databases. So that's a lot of gene space that is not known. And that's something going forward that would be there's a lot of value in figuring out what that is. So it was easier to show it like this, so it's more evenly matched, because the other way, just showing reads, there's going to be a lot of fluctuation in the peaks. So this is much visually better to see. And the other thing with this is, you'll see where I got this arrow here. This was really caught us by surprise that one class of bacteria, the Acinetobacteria, are 20 to 50% of the reads in these Brazilian samples. And we're like, what's going on here? That seems like this diversity is really skewed there. But I found a paper recently, thanks to a meeting I was at last fall at Duke, that these guys in China, Zhang et al., found in a soil biochemistry journal that soils in China below pH 6, they see the same kind of distribution in this. Oops, back that up. In the same taxa. And here we have the samples from the United States across there. And again, I'm using the Acinetobacteria. You see I do this a lot, right? And this is the same area that we're looking at in the Brazil. And you can see that they're only looking at 10 to 15% of the reads are in this class. And for the European samples, that class has been shifted up here. And again, you're actually seeing a little bit less, except for one outlier here, what looks like is around 20%, but they're about 5 to 10%. So you're seeing a much more even distribution across the different classes in the U.S. and European soils versus the Brazilian. And this is a phylogenetic tree of all the samples, again, at the class level. The Brazilian are the green, the European are the purple, and the United States are the blue. And you can just see how the Brazil samples are all being pushed down at the bottom of the tree because of that one class of bacteria, making it very, very divergent. You can see a few outliers like this. We expect a lot more of that kind of stuff. You see how the U.S. and the Brazil, and the European ones are all kind of mixed together there. And this is a nice slide because this shows all the samples in one view, and the extreme difference between the Brazil samples here and the U.S. and European samples. It's much more dynamic here when you can see how this distribution here. It's just sort of amazing that you can have one population of bacteria that can be half the reads in that sample. And so conclusions. The European and U.S. soils showed similarity to each other compared to the Brazilian soils. And determining what might be responsible for fast AI degradation, will be a challenge considering diversity of the U.S. and European compared to the Brazilian. And this study was used to support an EPA submission. And getting back to what Ignacio said earlier about the biomarkers, that would be the ideal case for Syngenta in a field. They go out, they take a sample, they extract it out, and they quickly can know, is that field, at that farmer's field, is that herbicide, is that insecticide, or whatever, is that going to be hanging around for a long time, or is it going to be broken down really fast? And those are the kind of things that would be very valuable going on just for even for submissions to different agencies for approval and things like that, being able to say we can track how fast things are going away across different regions and things. And thank you very much.